So like my hosts for the day said it. They said it all. I'm Herman Mwanja, and I work with the Fleming Fund Country Grant to the Infectious Disease Institute. So Anastasia, I can share my screen. Hope you can see it.
Yes, we can.
Yes. So did you ask that I can go ahead, delve into the session straight?
Yes, please.
Okay, thank you. So for today's session, or for the webinar session today, we're going to focus mainly on the environmental intersection of AMR. That is that intersection between the humans, the animals, and the environment itself, the plants and everything else. So the presentation, it's going to be really a brief one, though later on I'll ask us to just have a discussion to talk about these things. So the first one will discuss the intersection between AMR and the environment, then the drivers of the AMR. Then we'll look at those interlinkages that are really critical, then the implication of the AMR in the environment, and then the efforts to curb the burden or the role of the environment experts. So whatever is in the presentation has been preempted by a video that Daniel played at the start of the session for the team that was on before time. So I'm just going to build onto that video because it was really a rich one. So the environment is very critical in the global spread, proliferation, amplification, and emergence of the clinically relevant antimicrobial resistance that is within the bugs themselves, or what we call the superbugs. This is because it acts as a collection vessel for these bugs, and the sources of these bugs are the animals and the humans. But at the same time, the same environment acts as a reservoir for those natural resistance markers. And these markers could either be the bugs or other selective agents. The environment also facilitates the spread of these resistant bugs and the resistance markers from one place to another. So for this presentation, resistance markers, we mainly mean the antimicrobial resistance genes, the motile genetic elements, the selective agents, and other compounds within the environment. Then also at the same time, the environment offers that direct and indirect exposure route for humans and animals to this AMR. So the question most of us now are asking ourselves, what are those major sources, or what are the key drivers of AMR within the environment? So I tried to summarize these, and at least for each, when I was preparing the slides, I tried to ground them into scientifically relevant or scientific research that has already been done by international bodies, mainly UNEP. So the most critical driver of AMR within the environment is the poor sanitation, the sewage and waste effluent. And this is really because if in case there is really poor sanitation, people end up using antimicrobials irrationally. How? The poor wash conditions lead to people getting infections that they would have prevented. So they end up overusing or irrationally using antimicrobials. And as you know, that 70% of all the antibiotics or antimicrobials that we use in humans or animals are excreted. They end up in excreta. So if that excreta is not managed well, it all ends up in the environment, thus, leading to the emergence and re-emergence of this resistance within the bugs in the environment. There's also an issue of the effluent from the septic tanks. As you know, many of us on the call, or many of us within Africa, we are in settings whereby they're really low income settings, whereby we have waste being managed either on-site or not managed at all. I think I'll explain that later on as we move on. Then another point or major source of this environmental release is usually the leaching from open waste dumps. I know most of us have open dumps or landfills where we dispose of this waste. Then at the same time, also the urban runoff is also critical as a major source of this environmental release. Then another category we can look at is the effluent and waste from pharmaceutical manufacturing companies. So the high concentrations of the antimicrobials within the untreated effluent really drives or leads to the selection or evolution of the different bugs within the environment, making them resistant. This is because these bugs are exposed to minimal levels of the antimicrobial that wouldn't kill them, thus making them resistant to these drugs later on. Something we are going to also look at as we move ahead. Then the effluent from pharmaceutical companies also has some residual antimicrobials within the solid wastes that are also discharged. And also at the same time, it has the resistant microbes. Then another category or another key driver is the effluent from the waste, the effluent and waste from healthcare facilities. I think this is really straightforward. You know, everyone who goes, most of the people that are in these hospitals are really sick. So if they're being given all these different kinds of antibiotics and they're releasing them along with the resistant bugs from their bodies, and these bugs have been found to be resistant even when they're in the environment. An interesting case is where I can give you a scenario in Uganda where we managed to pick a sample from waste from waste within a hospital. When we processed it, we were shocked that we got okay, it wasn't really shock because we expected it, but we found bugs that were really resistant, and they showed resistance to several categories of antibiotics, including ciprofloxacin, ampicillin, and there were really high levels of resistance. And you imagine if those bugs are meandering around within the environment. Then another category is the use of antimicrobials and manure in crop production. These are usually the fungicides, herbicides, and and all those that are used. They really drive the resistance within the environment, the drugs themselves. Then also the manure from the animals, which is usually untreated, has the antimicrobial residues, but sometimes has the resistant bugs. And then the inappropriate disposal of all this is the one that is really leading to the proliferation of this AMR within the environment. Then also another category we can look at is the release of effluent and waste in the animal production. The other one was in the crop production. Now when you go down into the animal production and look at the aquaculture, those antibiotic resistant genes, the resistant microbes are all, it's really a whole complex issue that's happening within these different categories. And you know that now when you look at all these different categories I've talked about, one critical thing that you realize is, or one critical key driver is the irrational use of antimicrobials within the human and animals. Then the second will be the poor disposal or poor waste management within the humans, and that is really driving this AMR within the environment. That's where it is really a pivot. Those are the two pivotal points which we can now scale further to break down into the different categories. So maybe for whatever was in the table shown earlier, this is one of the schematics I picked from UNEP, from a document provided by UNEP. And it is basically the antimicrobial resistance in the environment. You realize that I don't know, can someone confirm whether they can see the pointer?
Yes, it's
Yes,
visible.
we can.
Okay, thank you. So realize that within the human population, there is a high consumption of antimicrobials since the 2000s. But the question is, do all these antimicrobials get broken down before they are released into the environment? No. So the antimicrobials are released either in their active forms or in their different broken down forms, which can still drive, or which are still selective when it comes to AMR. And then when you look at that up to 75% of antibiotics that are used in aquaculture are lost into the surrounding environment. I mean, the fish are given antibiotics either in the feeds or directly into their water. By all means, those antibiotics are going to end up in the environment. Then 70% of antibiotics globally are used actually in animals. But at the same time, over 70% of those antibiotics are excreted by these same animals, which are given these antibiotics or antimicrobials. So you're looking at this looming issue whereby it is this irrational use that's really driving this resistance. Then you look at the issue of the antimicrobial use in the livestock. This is said to jump up by over 67% by 2030. This is due to the intensification of the So that means if it jumps up by over 67%, that means there's going to be an increase in what is being released into the environment, the resistant genes, the resistant microbes, and the residues. So that is all the havoc that we're looking at that's going to be in the environment from the animals. Then when you scale down within the human populations, I know all of us in our countries, we have the wastewater treatment plants, but these plants cannot remove all antibiotics and the resistant bacteria. So by all means, all this ends up in the environment, even when we have these conventional treatment plants undertaking the treatment, still the waste from those plants still has all the effluent from those plants, which we claim to be treated water, is still not free of the antibiotics and the resistant bacteria. So when you look at now the human context, when you look at the healthcare side, antibiotic use within the humans we use about 30% of these antibiotics, but like I said earlier, over 70% is excreted of this 30%. So all that is ending up in the environment. We have septic tanks, we have sewer systems, but all these at a given point break down, and all this waste somehow ends up within the environment. Then you realize that also within the human setting, we have the pharmaceutical companies. They produce so many antibiotics every single day. And even up to of late, people are undertaking research to produce more antibiotics or antimicrobials. But the question is, where is this effluent? Does that effluent contain no antibiotic at all or residue? That is a question that you'll have to have in mind. So pharmaceutical waste, if untreated, by all means has chemicals, not only just the pharmaceutical compounds, that is the antimicrobials, it also has heavy metals. It may have detergent. All these things drive resistance within the environment. So so now the most critical part that I wanted to concentrate your mind on is the last two circles that I'm trying to flip between. Antimicrobial concentrations in most effluents are too low to be lethal to expose bacteria, but they are sufficient or may be sufficient to induce antimicrobial resistance. So this is something that is very critical. And at the same time, the multi-drug resistant bacteria are prevalent in marine waters and sediments in close proximity to aquaculture, industrial and municipal discharges. We have undertaken some sampling in Uganda, we have taken samples from the lake, samples from the wastewater treatment plant, samples from the sewer system, just to see whether this scientific evidence is actually real. But the thing is, these bugs are really multi-drug resistant. We found an enterococcus bug that was resistant to erythromycin, vancomycin, and it was 100% resistant to all the 5 antibiotics we set, including chloramphenicol. You know, so that is something that we have found out so far. So maybe to go ahead and now just delve deeper or narrow down further into these issues, into the interlinkages of the environment, the humans, and the animals with regard to AMR. So realize that, or maybe from the key, before we delve any deeper, this symbol here, I don't know what I can call it, but it's the one that is for the resistant bacteria. Then this will be for the residues, then the activities, and then the environmental aspects. So if I'm to start with the activities that are really driving the release of antibiotics and the resistant bugs into the environment, into the water, the soil, the air, is basically antibiotic production, the clinical environment, that is the human side, the aquaculture, dredging, landfilling, sewage or wastewater treatment plants, the livestock farms, domestic animal raise, so all these are the ones that are really the activities that are really critical with regard to AMR within the environment. But at the same time, humans are getting exposed from these different bugs and residues that have been deposited within the environment. That is to say, through the livestock, humans get infected, or they get in contact with those resistant bugs. Then the sewage, when the sewage system breaks down, chances are high that humans will get exposed. Then feeding of the animals, livestock and all those. So this is basically broadening the table that I presented earlier on the drivers of AMR. So maybe to put these interlinkages into context, like I said, I don't know whether all of us are from low resource limited settings or low income countries where some of these are not new. This is a community in Uganda where a sewer line is going through a community. This is a sewer line, but these communities here do not have actually access to this sewer line. They actually manage their waste on site. And this community had or has such poorly built infrastructure whereby the waste that's coming from humans, animals is within the environment. But at the same time, we have this water or the water distribution grid, that is the clean water. The pipes are within actually dirty water or in sewage, In case this pipe breaks down, by all means, there's going to be now there's going to be contamination of the drinking water. Then when you look at on the left bottom, a child is in a landfill, which is near a swamp. So, all those things. Then I think also this is not any different within settings whereby we have people fetching water from rivers and streams to use for domestic use. So like I said earlier, just wanted to describe a bit, but I'm not going into any much detail at this point, because when I was being given the terms of reference for the webinar, I didn't have to go into this so much, but to just give you an idea, assuming this pipe is waste from a pharmaceutical company, waste from a home, waste from anywhere, It could have all these, it could have antibiotic residues, it could have biocides or chemicals, with all these different selective agents, and even at the same time, it could have the bugs themselves. So what could happen is that within the environment, there could be natural selection. The bugs themselves could become resistant on their own because of the selective pressure. I mean, they have to survive. I mean, the jungle is survival for the fittest. But at the same time, we could have these different selective agents driving AMR, where they pose a selective pressure from these residues. And then it leads to the evolution selection for resistance within these bacteria. But at the same time, this bacteria ends up developing multi-resistance to such substances and transfer the multi-resistance genes to the non-resistant bacteria, that is horizontally, or to the next generation, that is vertically. So I'll still now delve back to the video that Daniel shared earlier. He made it very clear that antimicrobials are becoming very ineffective, to the treatment of these different illnesses in the human and animal populations. So, and this is not only in the human and animal, but it also goes into the plants, people are failing to undertake the different, is it and all those things. And then within that same video, it showed clearly that so many people are dying because of these resistant infections. And it is estimated that over 10 million people will be dying by 2050 due to these resistant bugs. And antimicrobial resistance really endangers the achievement of the SDGs. But now let's look at the implication of the AMR in the environment. Critically, now the AMR within the environment. So the residues and resistant bugs persist for really an extended time in the different environment compartments, even when the antimicrobial use has ceased within the humans and animal populations. And this, what happens is that it contributes to the presence of so many of these functional genes of the resistant bacteria within the human and animal waste, within landfills, within the leachate, in water, within soil, and within sediments. So when this is happening, that means there will be now further emergence and selection for new AMR traits. So what happens when now there's so many AMR or resistant bugs within the environment? There will be infection of the humans by these resistant pathogens. I'm sure most of us have seen situations whereby we have someone, or we know someone who has been diagnosed of having Salmonella typhi that is really resistant to so many bugs, but we're not certain of the source. I mean, it could be the environment, it could be, water that they drank. It could be maybe food they ate, where it was already contaminated by those bugs. So when the bugs get within the body, they lead to that colonization within the humans, and then that colonization within the humans of these resistant bugs can be a source for those genes, for the normal flora, leading to the horizontal gene transfer, which now in the long run leads to an increase in the hospital time, leads to an increase in the cost of care, within the human population. But as I explain all this, critical gaps actually still exist on AMR in the environment. I think this is where the biggest critical point of the webinar, in terms of reference, was in my thinking. So, while actionable evidence of the importance of the environmental dimensions of AMR has been mounting, some key data and knowledge gaps still remain. And these are really on the interlinkages and negative feedback loops that increase AMR proliferation in the environment. The global burden of AMR within the environment is still not known, though studies or action is underway to establish this, the AMR specifically within the environment. Then also the relative importance of each pollution source is really its nature and the magnitude hasn't been quantified yet. And then the type, quantity, and dynamics of environmental releases of chemicals and biological pollutants that affect AMR development, the transmission and spread in local environments or specific conditions is also not clearly known. Then the comprehensive understanding of the concentrations of the selective agents, that is the antimicrobials within the environment, the metals within the environment, their actual quantities and the number, is really, it's not something that has been comprehensively understood with regard to pollution of the environment and the role they play in the selection, co-selection and maintenance of AMR within the environment. So collectively, as this information becomes available, it can be used to prioritize interventions to mitigate AMR development and spread within the environment. So now based on those gaps, efforts to curb this AMR within the environment can be or are already underway, or new efforts can be developed depending on the context. So AMR, curbing AMR within the environment is really a multi-stakeholder issue, or what really is termed as of late as a One Health issue, which requires people from different fields. These people from these different fields would bring along their different ranges of new strategies and technologies, that are tailored really to specific sectors, as well as financial or business or climate or cultural contexts. But as you're doing this, we cannot forget the issues of the regulatory, economic, and in some instances, the cultural barriers. So thus far, or as of 2022, solutions exist. Concerted efforts by international organizations have been put forward. National action plans have been designed. Global action plans have been designed. And when you look at all these different plans and actions that have been designed, those that are specific to the environment look at the prevention and mitigation actions for promotion of sustainable production and consumption, to establishing ambitious targets, and setting out clear visions for the global roadmap. So now I'll just this early this year, the United Environment Program, that's UNEP, provided or published a brief to policymakers, where they were looking at the environmental dimension of AMR. So these are some of the critical roles of the different environment experts that I found will be relevant to the team on the call. Enhancement of environmental governance by developing and enforcing environmental regulations related to antimicrobial manufacturing, water, sanitation and hygiene standards, agriculture standards, solid waste management, and infrastructure. So this is really through, developing the legislations, the codes of good practice, policy guidance, to really support action to reduce and minimize environmental releases of AMR, relevant pollutants. And as these are being designed, one critical thing that should not be forgotten is the issue of stewardship, and then it can also be through the strengthening of research and innovation frameworks, transparency, accountability systems that allow consumers hold producers accountable to prudent antimicrobial use. Then another critical role is to be able to identify and target priority AMR relevant pollutants. This is through reduction of the releases of chemical and biological pollutants affecting the AMR in the environment, and also address their origins. Then ensuring that wastewater is treated and managed at source so as to reduce the number of antimicrobial resistant bugs and the gene concentrations within the environment. But as this is being done, also the clean water has to be treated before consumption by the humans. There should be access to clean water, by everyone. And by access to clean water, we mean now we need to go a step further. I don't know whether on the call we have someone who works in an environment lab. In our labs, we do microbiology, but we only stop on being able to qualitatively screen for E. coli and total coliforms. But we need to go ahead and an extra step and be able to say that, yes, we are saying that it's within the required range of, because some provisions, when you read, given a country's context, they may say for water to be safe, there should be about less than one coliform forming unit. But I mean that one coliform forming unit may be the one to bring havoc. You may not know what coliform it is. What if it's an enterococcus and a pseudomonas bug that's really resistant when it gets into the human body? Then there's also need to ensure that the environmental application of antimicrobials in plant-based food production systems is minimized through measures such as the integrated pest management, which I'm sure many of us are well versed with. Then environmental experts or environment experts need to improve the reporting, surveillance, and monitoring of this AMR. They need to measure the impact of the antimicrobial pollution on biodiversity and integrate this with the environmental monitoring data. As you know, in all countries, there are standards that have been set for monitoring the environment or pollution within the environment. That is surface water, solid waste, groundwater, and even air itself. And then there's also a need to do risk profiling to establish how likely AMR relevant pollution occurs through surveillance methodologies based on other monitoring systems and approaches standardized around the world. Then there's also a need to develop and strengthen systems for transparent and swift collection and reporting of production, sales, use, and disposal of unused or expired antimicrobials. I'm sure many of us are asking ourselves, why would we do the surveillance and monitoring? Surveillance and monitoring provides us with that continuous information, or helps us increase the knowledge base that we have on AMR within the environment. When we have this increased knowledge base, we can now go into issues of prioritizing financing, innovation and capacity development. When you have that data, you can now be able to more confidently enhance those private public sector collaborations, realignment of incentives, and then you can still also be able to provide a clear business case for investment, including the investment of domestic resources in cost-saving or alternative technological approaches that reduce environmental AMR risks. And this, like I said earlier, the reference to these roles is given below here. Thank you, and thank you for your time. And over to you, Daniel and Anastasia. Thank you.
Yeah, thank you. Thank you so much for that wonderful presentation, Mr. Herman. It has really been, a good Kickstarter for us in the environment scene, and I hope that we've all also learned something and also identified some gaps, some potential innovations, and some of the roles that we can also play. This juncture, maybe if you have a question, you can type it in the chat box or share it or raise your hand, then I think we can proceed with the question. Yeah. So anyone? I can see Colonel, your hand is up. Maybe you can go ahead and ask a question.
Okay, thank you so much. Am I audible?
Yes, you're audible. Can hear you.
Okay, thank you once again. My question is rooted from an environmental analogy of pollution from gases. we used to talk about the residence time of a pollutant once it is released to the environment, and that residence time having an effect on the magnitude or the extent of damage it would cause to the environment. So in context with our own antimicrobial resistance, we are saying that antimicrobials are being released in the environment. Some of them have stable structures, some of them have less stable structures. So I wish to understand if we have defined residence times for antimicrobials that have been released to the environment and the extent to which that can have effects to our to microbes or our environment in general from that residence time effect. Thank you.
Thank you very much, Kanu. Hope that's the right way of pronouncing that name for that. So based on the research that, thank you very much. I don't know, am I loud and clear?
Yes.
Yes, we can hear you.
Okay, thank you very much, Kanu, for that. Hope that's the right pronunciation of that name. So with regard to residence time, not like I said, those are some of the gaps that we need to address so as to increase the available knowledge on AMR within the environment. As you realize, it is just this year that the tripartite plus, or that UNEP joined the tripartite organizations, that is the FAO, OIE, and WHO. So you realize that we are still lagging behind with regard to many of the scientific literature of AMR within the environment. Many people have concentrated on the genes, but with regard to the residence time and AMR, the residence time of the pollutants vis-a-vis AMR, I can't say I've come across any literature of late. Thank you.
Okay, thank you so much.
Thank you so much, Kanu, for that question. I think you've also identified a gap there. Maybe that calls, you can think of how you can do something to intervene on the same. We can head over to Iradat. You can maybe unmute and ask your question, kindly.
Good evening, everyone. Am I audible?
Yes, we can hear you.
Hello, okay. Okay, my question is about the waste management system. Like you said, that there are some part of the country that don't have waste management. So I want to ask about maybe in a rural area where they don't have a waste management system. So how do we communicate with these people on how to actually manage their waste so as to reduce the environmental factors that might lead to AMR?
Well, thank you very much for that question. So, the way to communicate with these people, it's, in my perspective, it's dependent on what waste management, what waste you're looking at. If you're looking at solid waste management, that is anything away from human waste, you'll use a different communication plan. If that's what you meant. If it's liquid, it will still be different. An example is the teams in Uganda that are doing the community CLTS, community-led total sanitation, where they're trying to do it with the open defecation. So those strategies are really, all the different strategies for that communication are really out there. And it really depends on what waste you're looking at. If I'm looking at animal waste, I mean, I'll have to tailor it in a specific way. Look at first get the context that I'm looking at, then be able to communicate whichever message I'm anticipating to communicate. Thank you.
Thank you so much.
Thank you, sir.
Okay, I hope your data is clear. Maybe we can move over now to the chat box. There are some questions, then we come back to those who raised their hands. And the first question is from Masa Mese, Winnie. And so she thanks our speaker for the presentation. So I'll just read it as she has shared. So my question is there any proper way of antimicrobial disposal? Because everything almost seems like a vicious cycle, because no matter what is done, these antibiotics will go back to the environment, either through air, soil, or water. Thank you.
So thank you very much, Winnie, for this question. At least, given the kind of team that is on the call, I'll just pose this question back to you. What is that innovative way, or that way you can think about to see that this vicious cycle is broken? You know, yes, we are very sure that it all goes back to the environment, and we have stewardship programs being undertaken in the human and animal sectors. But the question is, what can be done in the environment sector to see that this is really minimized? Because right now, the burden is already here with us. So we cannot say we are going to deal with it, but we can only minimize it. So for that question, I don't think I personally have an answer, but I'll just pose that as my view.
Thank you so much, Mr. Herman, for that. I think this reiterates also rhymes with Mwitupa's question. So I think this question is just posed back to the group, and I think this is also an opportunity of we can also try and see how we can address some of these challenges in our own context. We'll move over to Telespo. Maybe you can unmute and share your question.
Okay, thank you. Can you hear me?
Yes, we can hear you.
Okay, thank you. Thank you, sir, for good presentation. Based on what you have presented, I found that the natural environment can be like an important reservoir of antimicrobial resistance, especially when you try to see the most of the drug-resistant microbes, which are highly found in the environment, like in water bodies, in the soil, and in air, which can be exposed to those antimicrobials. So my question looks like this: when you try to see, you found that the environmental dimension of antimicrobial resistance has received comparatively less focus when you compare to the antimicrobial resistance in humans and animals. So like you as an expert, I wanted to hear your view. What do you think, and what do you think the environmental dimension of AMR is given this focus compared to the humans and animals? And what do you suggest that can be done?
Thank you very much for that question. Guess you meant that why is the environmental dimension given less focus, right?
Yes.
Okay. So in my thinking, it all goes back to health. You know, the definition of health. Regardless of whatever we do, the health of humans comes first. So the experts or people who design these programs, they'll obviously look at the human sector first, regardless of whichever other sector. Then they'll see how do we now bring on. Then as literature comes on, or as more research is done, and then they realize that, well, this problem is not only actually in the humans, animals also, you know, could be the cause. We consume animals, or we consume food from animals. So they went ahead and then moved into the animal sector. Then, you know, then they realized that it's actually beyond the animal sector and human sector. And then they pushed into the environment sector. That's from the policymakers' view. But again, when you go down, when you delve now back a bit into, and you go back into the academic world or into the world of academia, you realize that even when we study, us who did environment health or environment studies, I mean, some of these issues are not anywhere. You know, we don't study them. So it all comes back to the different, what can I call them, syllabuses or syllabi or curricula that we studied back in school. So by the time, if we are producing professionals who are not tailored towards some of these aspects, it's really hard for a specific area to get recognition, as if I'm to call it that. And then the way I feel like what we need to do jointly is to, first of all, always collaborations with both the local and regional and international bodies. Now we are lucky that UNEP finally joined the tripartite. They signed an MOU. So now, in UNEP's plan, I don't know whether some of you have got a chance to look at the United Environment Program plan for 2020 to 2025. Environment is one of the things that after climate change, AMR is one of the most critical aspects they've vied or committed to undertake or to curb. So I think collaborations, both national within our countries, then inter-country within the different countries, are all very critical, just the way it's being done in the human sector. Thank you.
Thank you so much for that. I hope for your question has been addressed, and just to think brings in the issue of multidisciplinary approach, right, and also training right from early education, and especially in our tertiary level institutions. Maybe we can now go to Zachariah. Your hand is raised. Maybe you can unmute and maybe ask your question.
Yeah, thank you very much for giving me the chance to speak. Can you hear me?
Yes, we can hear you.
Okay, thank you very much. Thank you very much for this wonderful presentation. I was thinking in some literatures we normally see that human waste in sense of urine, feces, and the likes are being passed. As when you take antimicrobials, those antimicrobials are seldomly metabolized in the body, and some residues, some metabolites are being passed to the environment, like through human waste. Like normally when you pass your waste as humans, you pass some metabolites. So my question here is that are there some mechanisms that could be used to kind of used to fully synthesize those metabolites in the body, so that when you're passing your waste as humans, you're not passing residues of antimicrobials to the environment, which will lead to the rise of antimicrobial resistance in the environment. I hope my question is passed. Thank you very much.
Okay, thank you very much, sir, for that very interesting question. In my perspective, I feel like I cannot say whether I know any interventions that can be done, but one thing I can say is to really prevent the irrational use of antibiotics, or to undertake those stewardship programs. Very well, I can give you a scenario. In case any of us got, or in case many of us get upset stomachs, someone usually runs in for an antibiotic, as someone, even without diagnostic or microbiology laboratory diagnostics, people are running into using these antimicrobials. So all I can say is we just need to reduce the amount of antimicrobials that we consume in the human sector, so that we can be able to at least, or be sure that we are releasing minimal amounts into the environment. I mean, if I didn't take an antibiotic in the last 1 year, I'm certain that I will not release antibiotics into the environment in let me say, in the last 6 months, and I will be very certain. That's my view. That's my perspective.
Zachariah, are you there? I hope that's okay.
Yeah, thank you very much. That's very fine. Thank you.
Okay. Yeah, so juncture, I'd like to combine two questions which really touch us, especially as from the low and middle income countries, because agriculture is our backbone, how we grow our crops. And that's a question from Anita and Idris. So Anita asks, how can we tackle the AMR linked to manure usage? Yet we know that most of the world based on agricultural activities depends on manure. And I'll just like to combine this with Idris Tajudin's question, which is, as we know, many countries from Africa are low income countries, and many use animal fecal matter as manure, and this can contribute to increase in AMR rate. How can this situation be handled, as many farmers actually lack capacity to buy ready-made fertilizer, and it's also not easy to convince them to stop using this, as they also look towards maximum production? Thank
you. Thank you very much for these great questions. So, well, tackling the AMR linked to manure usage, it's really, I can't say I have a definite answer for this, but what I would think is we should be able to either use other substitutes to this manure. I mean, instead of using the manure from, because this manure, it could be animal or human based, So we could either opt for other options and either use the plants themselves as manure, but the AMR linked to the manure, the only way is either we handle it well so that there's no crossover into the humans by all means, or handle it in the best way that there's no crossover or there's no spillover into the human population or into those on animals that stay in water. So we just have to find a way for this. We just have to find a way of handling it with the utmost care, because like the next person posed the question that we are in a low limited, in a low resource setting whereby we can't access fertilizers. I mean, people are poor, so people have their best option is the manure. So as we're using the manure, we just have to handle it with care. As you know, most farmers just handle this manure as is. Like, you'll find someone touching dung. I mean, we've seen this in farms. Someone is picking out dung with their bare hands. So we just have to find a way, whether we design standard operating procedures, or we design, standard ways or good practices, the codes of good practice when you're doing many of this agricultural work. I mean, we need now to move the biosafety, biosecurity away from the lab now into the farms. It's also something we can look into, so that we, as you know, at the end of the day, we are protecting the health of the human, regardless of whatever we are doing. And how we shall protect the health of the human is by ensuring that the environment is safe and the humans are also safe and the animals are also safe. So that's my take on that, or perspective.
Thank you so much for that response. Yeah, and maybe just what a bit, I think there's a project that is ongoing in Tanzania that I think they're trying to explore some ways of treating manure, especially from the poultry. I think it's funded by ICAS. So it's something that I'll share the link, then we can try and read on the same. And I think one of us has a direct link to the project officer. I think that's Mabula and Professor Mdegela from Sokoine University. Yeah, so at this juncture, maybe we can just take the last question because of time. And I believe we'll have another session next Saturday, maybe just to talk more, especially on the pharmaceutical industry and the pharmaceutical pollutions. Yeah, so for the last question, we can just take it from Ellie, then maybe Benny, we can address your question in the next session. And so Ellie asks, thanks for this amazing question. My question, on one of the slides, you've established that our current water purification and sanitation treatment are not able to fully eliminate antimicrobial residues and the resistant bugs? Are there mechanisms that we can introduce to optimize the current water treatment systems to further reduce antimicrobials in our treated water? Thank
you. Thank you very much, Ellie, for this question. So the mechanisms that can be introduced is we just have to look at those mechanisms that, one, totally kill off these bugs, and two, that totally screen out the residues. But as you know, we shall go back into the context, or we should be cognizant of the context that we stay in, the local context. I mean, if we are in a low resource setting and we're saying we want to have all these, what is the investment vis-a-vis the output? Because you realize that even when this water, or I'll just give you my view, you realize that even when this water is treated, it's going to end up back into the environment. Like in Uganda, I can give you an example. When the wastewater is treated in a conventional treatment plant, it is sent back into a channel, an open channel, which I can call a stream or a river. But when you look at this stream down or downstream of this channel, other people are connecting their effluent pipes to this same channel. And when you follow the channel down to where it disposes the water into the lake, you're going to still find contamination along the way. So in this sense, the wastewater treatment plants actually I've now come to understand over the years that they are meant to reduce the load of the resistant bugs or the load of whatever waste is in there, so that the water goes into the environment in acceptable levels. But these acceptable levels, when you look at the AMR dimension, they're actually not acceptable levels, because these bugs are resistant. These bugs have done, so much has happened to them. The stable elements are still there. The genetic genes are still there. So it's something whereby we need to look at the investment vis-a-vis the output. Are we ready to go into UV or ultraviolet technologies whereby we want to kill off these things fully, chemical treatment, so that we are sure of that we are producing clean water that you can drink from a wastewater treatment plant? And then at the end of the day, am I sure that this water is going to go back to the environment, or is it going into consumption? So you just have to weigh that. And for now, I can't say that there is an alternative that I can say can do away totally with the resistant bugs and the metabolites within that water. It's not any that I've seen yet. Thank you.
Thank you so much, Mr. Herman, for that really wonderful session. We really do appreciate for being with us today and for taking us through, and really for the sacrifice, taking us through, especially being on a weekend evening. I'd also love to thank in absentia Grace Lubega and Dr. Musoke, both from Makerere University, also helping in coordination of the webinars. I believe we'll have another session next Saturday. Maybe Herman, you can confirm?
Well, maybe Daniel, we'll keep in touch. Okay. At least I can say, yeah, for the group, at least I can say I'll try to fix time for next Saturday. But at least about next Saturday, we shall just have discussions about these issues. And one critical thing that came out clearly was the word innovations. And I love innovations personally. So I think we can discuss those. We can keep in touch, discuss those, and maybe we can, we'll be able to deliver another session next Saturday.
Yes. Thank you so much. Yeah. And I believe this is a challenge to all of us. I believe we've seen some of the gaps, and I think the environment really it's something that we can relate to in the day-to-day, in our day-to-day lives, especially since most of us also from low and middle income countries. So these are challenges that we experience every day, and there are things that we have seen and we can relate. Yeah, and the good thing I think with the environment is because it has a face, you know, unlike maybe resistance in human health, where it's kind of hard to see. You know, we're able to see pollution, and you can see some of the effects of this pollution. Yeah. So thank you so much, everyone, for the session. We do appreciate you, Mr. Herman, for really joining us today and for taking us through. I also like to appreciate my co-coordinator, Anastasia, and the whole team for making it to the session. I believe it has been a great session, and in case anyone didn't join, then I can share the recording with them. Thank you all, and I wish to maybe wish us a wonderful evening. Thank you so much, and have a wonderful weekend. Bye-bye.
Thank you.